Good evening, everyone. Today, we are having a very interesting session on cancer genetics, and the topic for today is genetics and colorectal cancer. Today, we are very fortunate to have Professor Rajiv Sarin, who is a professor in radiation oncology at ACTREC, which is a research wing of Tata Memorial Hospital Mumbai. Sir has been trained in clinical oncology from TMH Mumbai and later on from Royal Marsden Hospital London, and then he has his training on cancer genetics from University of Utah, US. Sir is one of the pioneer in this field of cancer genetics in this country, and he started his hereditary cancer registry dated back in 1996. Sir also has a dedicated cancer genetics lab at ACTREC, popularly and famously known as Sarin Lab, where many research work related to cancer and genetics are ongoing. Sir is also the lead investigator of International Cancer Genome Consortium and the Indian part. He has been author of multiple peer-reviewed high impact factor articles in many national and international journals. He has been part of editorial board of multiple famous international cancer-related journals, including Lancet Oncology. And it is our fortune that today we got Sir here for teaching us about genetics and colorectal cancer. May I now request Sir to take over the session and please start your presentation. Thank you so much, Sir, for being with us.
Okay, so thank you very much. Thank you for inviting me. And whenever I get a chance, unless until I'm doing something terribly important, I am always ready to talk about cancer genetics, especially the genetics of cancer. And the reason why I'm always keen to talk about it, because I know how ignorant I was and how long it has taken for me to get basic understanding of the genetics of cancer. It shouldn't have taken so long for me to understand, but that is a reality. That's a real life story. I don't want others to take that much time to understand genetics of cancer as I have taken. And dealing with now almost 9,000 families, I have developed a perspective about the hereditary parts of cancer, like dealing with cancer patients. All oncologists develop a perspective about cancer. All of it is not described in textbook. Textbooks describe only few things. So similarly, 9,000 families means 20, 30 or 40,000 individuals who are related to each other, and they have fears, they have joys, they have many things. So my perspective. And as Dr. Mandal said, I wear two hats. I was a hardcore oncologist, radiation oncologist, and then I turned into a metamorphosis into oncologist come clinical and laboratory genetics. So first and foremost, is it of clinical relevance or just academics? Because I believe many people here are students. So is it only because sorry, I've written GI cancer. So only because it may be asked in an exam, it could be a short note or something. These days, genetics is people talk about it. So is it of clinical relevance? So 10 to 20% of GI cancers, I've written GU, will have genetic alteration in the germline, which is inherited on the tumor tissue, which is somatic, which could explain why they developed a particular cancer at a young age, their future cancer risks and surveillance for second primaries, if their close relatives are at risk for inherited cancer, if their tumors would respond to a particular type of cytotoxic or targeted or immune therapy. So all these, so it has come not only for risk prediction, but also for precision medicine. And now there's a field known as precision prevention. Based on that, you can actually, prevent the cancer. So what practicing oncologists need to know about it? I'm not going to give you a long lecture about, genetics, or I'm not starting from Mendel. Of course, he's my guru, Mendel. So basically, practicing oncologists need to know to advise advise appropriate screening and treatment for their patients, better with inherited cancer risk or having an actionable mutation. About the future, they can tell about the future of this patient, of the family, and also to some extent the future of the doctor and the hospital depends upon it, because in times to come, if you are genetically ignorant, it will affect your practice, and your hospital also would suffer, because many of these patients would want, they seek opinion and they'll go to different hospitals. So what practicing oncologists need to know, and the first point is when to suspect. Of course, every cancer would not have a genetic component or a hereditary component. Having said that, when I talk to the PhD students, first year, the JRFs, I tell them every cancer is genetic, true or false. And when I ask this question, they get a little flummoxed. Like, what does this mean? I said, there's only one answer, yes or no. Is every cancer genetic or not? No one gets it. Most people say no, every cancer is not genetic, and I emphasize every cancer. Then I say no, every cancer is genetic, because you can never have a cancer where all the genes and chromosomes are absolutely normal and the defect is only in the cell membrane or mitochondria or some Golgi apparatus or something else. So in the multi-step carcinogenesis process, if you remember, Weinberg and Hanahan and Weinberg in year 2000, they came with the hallmarks of cancers. So 8 hallmarks, and then in 2013 or 11, they came with another 2 hallmarks. One of the key enabling features for cancer cell to acquire all this is genomic instability and mutation in the genes. Unless until genes are mutated in a cancer cell, they would not have uncontrolled growth, they will not evade apoptosis, they will not be able to develop drug resistance and all those things. So every cancer is genetic, but only some cancers are hereditary. So when we say genetic, it has to be inherited. So there the mutation starts in the tumor cell because of the transformation. One cell becomes transformed, what we call malignant transformation, and the daughter cell will, and during transformation process, the genetic alterations have been acquired in that cell, which is the malignant transformed cells, and the daughter cells will inherit that, and then their daughter cells would inherit that. And over a period of time, the daughter cells or the granddaughter cells, great-granddaughter cells will have a new mutation, and a subclone of that tumor cancer would develop. And the subclone, there will be subclones of subclones. Some of them would acquire metastatic potential, and some of them would become more aggressive. Some may lose their original features, some may acquire a new feature. That is how a tumor evolves, tumor evolution. That's why recurrent and metastatic cancers are more difficult to treat, because they have acquired so many more gene mutations that they become more resistant to treatment, and they do not respond to what drugs they were responding earlier. So every cancer is genetic, but when the transmission of the mutation is not just to the daughter cell, tumor cell, but it has been transmitted from one generation to another. That means the tumor, the original mutation was a germ-line mutation. Germ cells means ova and sperm. So if there is a mutation in the germ line, it is transmitted to the next generation. It is not just the next tumor cell. So the germ line mutation makes it is inherited down the generation. That's what we typically call as the hereditary cancer. So when to suspect, history scans, and it is not just history. Although Indian doctors are supposed to be very smart, just within few minutes we get everything right. But one thing we don't do right is we don't take a proper history. We don't take a detailed history, and I don't blame ourselves completely. I mean, of course, partial blame will come on us, because the way we have been trained, majority of us have been trained in very busy public hospital. You would imagine the outpatient clinic is so heavy, whether it is a skin OPD or whether it is a surgery OPD or medicine, any OPD you take, you will have a huge waiting listing of patients, and at one point of time you are doing 100 things. So history, we very quickly, although when we read the books, Hutchison, it would say very important, the importance of history, but in real life, unless it's a long case where we are forced to take a long history of, or we're presenting a case, long history of 15-20 minutes, in real life, we just ask leading questions for that case, especially if you become experienced, and then move on to our examination finding, ordering some tests, interpreting tests. But for hereditary cancer, I think history is the most important thing. And I will share a little anecdote. You know, till about 90s, I did my MD from Tata Hospital in 91. I went to England. There's quite a famous hospital there known as Royal Marsden Hospital. I spent 4 or 5 years there. In 96, I came back and joined as a consultant in Tata Hospital, assistant professor and consultant. And when I went, I was a senior resident in Department of Radiation Oncology in Tata Hospital. And I think things have changed now, but in our days, senior residents in Tata Hospital, any department, we were the most arrogant people in the world. We had soul and we thought we knew everything. Actually, we didn't know everything, but we had seen almost everything from top to bottom, all stages. We controlled our consultant, we controlled the ward, we controlled the juniors, we controlled the whole world of Tata Hospital. And being in Tata Hospital, we were so busy, the whole world for us was Tata Hospital. So we went with that. And actually, I went with that mode, frame of mind, went to England. After 4 or 5 years, came back, had a fellowship degree and, some sterling pounds and a bank balance and everything else. And actually, I thought at that point of time, well, what I learned was there how to do things in a more organized way, a little bit of polish, a little bit of this and that. Rest, everything else we were already doing in Tata Hospital. And what I had not realized that I had picked up something very unique, which purely because of my stay in England for a few years, otherwise, the whole field of genetics. What happened in 96 when I became consultant, suddenly I found that every week I'm getting one patient or sometimes two patients who give a significant family history. I thought, what has happened in just 4 or 5 years? It's not that suddenly hereditary cancer has become more common in India. It was never so common. Previously, I would find one case in maybe few months who had a strong family history. I was curious. Then it occurred to me, I think just being in England, trying, like you said, when in Rome, be like Romans, trying to copy them, how they used to take history, because you have to take full history, record it, and someone else, you don't want to be embarrassed that you have missed this important history. So the way I was taking history had changed. And the whole thing started just with that, because the way I was taking history had changed. And that really brought about, I started, previously what my mind was not knowing, my eyes were not sort of seeing. Then suddenly I was started seeing. And then initially I got into it more from research interest. And then at that time, there were no genetics lab. BRCA1 and 2 had just been found. Most of the many genes had not been identified, or there was no genetic testing for them. In India, there was not a single genetic testing lab. I realized it is too, I've bitten too much. Then I can chew. I wanted to get out of it. But the first 100 families whom I had talked to them, and their first time these families had met a doctor who was actually listening to them. Otherwise, doctors, we just tell them, you do this, do that. Patient asks, what about my daughter? We just say, what we say, don't worry. Okay. So they had heard so many times, don't worry, doctors telling them, don't worry, but they had not stopped worrying because they had seen what is happening in their family. They had not stopped worrying. And that was the first time a young doctor, I was, 96. So you can imagine I was quite young. I'm still young at heart, but that time I was really very young. And someone who was actually talking to them, taking history, asking who was there, your mother's sister's daughter, your uncle's, elder son. So they just lashed on to me. And every time, so I used to tell them, you come after 6 months, we'll tell you. I was in the hope that certainly in 6 months time, I will know more, or I will get some resources that can help them. But that did not happen. I wanted to get out of it. I thought I am like stuck like Abhimanyu in the Chakravyuh. I have gone inside it, but I had no means to help these families. And no one wants to be embarrassed in front of patient again and again. Telling them the same thing after 6 months, after 6 months, after 6 months, after 6 months again. I had no answer. I mean, they had already told me the story. I had written pages and pages, and that I used to do in the lunchtime clinic, because in the routine, clinic, if you start spending 45 minutes with one patient, other patient will start become very edgy. And I'm talking of 96. We used to see even more number of patients. So then, one day the doctor within me that sort of took over from the researcher within me. I said, well, it's true that I can't do research because I don't have a lab. There is no lab in India which is doing research on genetics in 96, 97. I do not know, and without genetic testing, when I used to read the advice for cancer prevention was based on genetic testing, I can't do it. So I said, but at least I can take more interest, you can counsel them. So then from a researcher, I became a genetic counselor, but at least what are their questions, what is in their mind, what troubles them, based on my knowledge, whatever, and I'm a self-trained, self-taught cancer genetics person. So is to read and explain to them this is what we should do. And there would be occasional patient, 1 in 200, who had a relative in US, and there they had got the genetic testing done in 97, 98. Myriad Genetics that time was also charging more than $1,000, and there was nothing. But we made a familial cancer registry, and I have the register here. One second, I'll just show you. If I have the anyway, it's all right. So so many of the registrars who were my registrars, now they are senior consultants, Dr. Vinita Goyal, others, they helped me build up this thing. And then that's how now we have 9,000 families. So history is the most important. After you have taken the history, you should know how to put the history in a pedigree. Again, many of us have forgotten how to make the pedigree, what are the symbols that are used in the pedigree. All of us know males and females. My own pedigrees of 80, 96, when I look at them, I laugh at them. If a student makes a pedigree like that today, I would, give them a look. But that is how I also used to make pedigrees. And it's all, life is a process of learning. So I think we should not get worked up. And that's why I'm telling you this thing. Now, when to suspect, it is not just history. Now we know, based on the last 30 years of research, many times a scan will have a telltale sign which will indicate that this could be a genetic diagnosis. This could be a hereditary type of cancer. Many times pathology report or pathologist would make a description which is very classical indication that this could be a hereditary. Then examination findings, many phenotypic features you would uncover, and I think the phenotyping will be done only by the doctors are phenotypers, we are phenotypers, and the people in the lab are the genotypers that look at the lab. But genotyping will be done only if, as a doctor, if the phenotyper does his or her job properly, then only the phenotyping will come, genotyping will, otherwise it will just stop there. So when we suspect, based on our history of the patient gives, family history, the scan findings, histopathology or the examination findings. So for two reasons we have to suspect things. One is inherited cancer risk or a reasonable likelihood of actionable mutation. I think in the last few years we have gone into the field of precision medicine, and in precision medicine many times when you say precise, so the precision comes from knowing what is the actionable mutation, and it is generally a somatic mutation, but sometimes it is also a germline mutation based on which, like example of PARP inhibitors in ovarian cancer, based on which you can take action in the terms of giving a particular treatment to which a patient may respond. Once you have suspected, then what do we do next? Genetic counseling. Now, most of us, like when I started, there was no genetic counseling. And why I wanted to get out of this? Because I was so ill prepared to answer their questions. And they would ask very probing questions. Hey, that you had said this, what should my daughter do? What shall my son, he's only 9 years old, shall I do anything for my son or not? And I had my medical knowledge was also, medical clinical knowledge was also not very good, but still you could read it. But how to tell them, I wasn't sure how will this person would react, what I should say, what I shouldn't say, I had no clue. So when you have no idea, most of the time you will just keep quiet or you will give an evasive answer. So genetic counseling in 96, 97, I could do that. In year 2020, you cannot do that. If you try to do that, it really will reflect upon your professional efficiency. So, of course, like I'm also not a genetic sort of counselor, but most people would have to learn the basic things about genetic counseling. Like after you have taken a history, you have suspected a diagnosis, you are ordering a genetic test, or someone has already come with a genetic test. what are the essential things that you need to ask, you need to tell, what are the leading questions you need to ask, what are the things which will be open-ended, what not to say. You should know what are the common concerns of these families and how to address them. And again, I don't have slides for this, but I'm just telling you, especially it has come from ovarian cancer, because NCCN guidelines says that all high-grade serous ovarian cancer, which is majority of ovarian cancers, and most of them are in stage 3, that all high-grade serous ovarian cancer should have germline BRCA testing. So the workload has increased so much for genetic testing, and genetic testing has to be done after genetic counseling. Genetics clinic in the West, they are just unable to cope. I have had 2 patients in our genetics clinic who live in US who had come to India, must have come on a holiday, not only for this. They sought an appointment from us for genetic counseling, because I said I was in US, they were getting appointment after 7 months or 8 months. When the first patient said, I didn't believe it. I thought maybe some insurance problem. One more patient said that yes, they're getting appointment after a long time. She didn't say 8 months. And the reason is because there's no way suddenly the number of genetic tests have exploded. You cannot multiply, the genetic counselors will not grow exponentially. So there is a move known as mainstreaming of genetic counseling, and at the Royal Marsden Hospital, they have been doing some pioneering studies in that. And what happens in that the practicing oncologist would know that in which patient to order for genetic tests. So basic things they have to know. And if a mutation is found, then of course, then there are ramifications of so many germline mutations, then that patient is referred to the genetics clinic. Previously, every patient was first referred to genetics clinic, and then genetics clinic will decide and discuss whether genetic testing should be done or not. If genetic testing, majority 80-90% genetic test result will be negative, they'll be sent back to the oncology OPD, and the rest the genetics and combined follow. So in mainstreaming of genetic counseling, what is being done? The oncologist deals with the basic thing. Of course, someone has a significant family history, that family is sent to the genetics clinic first. In absence of a significant family history, the oncologist takes the basic history, record the findings, and order for genetic test, should know which genetic test to order. And then if a mutation is found or a variant of uncertain significance, then that patient is referred to the genetics clinic. So that makes the whole process more efficient. So genetic counseling and then genetic testing, again I explained in the beginning that the mutations could be a germline mutation, which is a mutation which is present because it was present in the germ cells, gametes, sperm or ova, it will be present in every cell of the body of that person. So that is one thing. But it could be a somatic mutation, means only in the tumor cell. Like in ovarian cancer, the question is whether you should test the FFP on the tumor block or on the blood, which is germ cells, which is germline mutation. So that again you have to know, or you should do both. Then how to interpret genetic test report and how to act upon. So most of us, as oncologists, my colleagues, they know, they have read NCCN guidelines, how to act upon genetic test report. But in between the process of interpreting genetic test report. So like on a PET scan also, when you get a FDG PET CT, initially we were not knowing how to interpret. I mean, initially I've seen all the things coming one by one. So even MRIs, we were not sure how to interpret. When we could distinguish between T1 and T2 and all that itself used to give us great pleasure. Oh, we can distinguish this is T2, this is diffusion weighted, all these different sequences. So genetic test report interpretation, you may not know everything, but basic things, because the patient will come to you with the genetics report, you have to know basic aspects of the genetic test report. That means whether you have to act upon it. Act upon it means you may have to counsel patient about family cancer risk, about further treatment, about future surveillance or prevention, or you may have to act upon it in the sense you may have to ask for more genetic tests, or you have to say nothing to worry. Okay, and most of you will not be lucky to have a genetics clinic in your hospital where you can just say, okay, your report has come, Mrs. Singh, you go to the cancer genetics clinic, they will explain this to you. So this is a previous picture. You can see in the background, I had made this. So I've changed for my another thing, I've changed this. So, mutated gene, these, again, I'm using this to show that I use this for counseling, that uh, the mutations in the cancer genes, germline mutations, are invariably they are heterozygous mutation. That means only one copy is mutated, because suppose chromosome 17, there is a pair of chromosome 17, one you got from your mother, one from the father. Maybe the one you got from the father, that has the BRCA1 mutation, not the one you got from the mother. So it will be heterozygous mutation, and the mutated gene will result in alteration in the protein. And when the proteins are altered, they either lose a function, which is in the tumor suppressor gene, or they gain a function, which is in the oncogene. Proto-oncogenes become oncogene, and the result in cancer. And depending which gene is mutated, you may develop brain tumor, retinoblastoma, breast cancer, thyroid cancer, GI cancer, endometrial cancer, sarcoma, soft tissue sarcoma, any cancer under the sun, depending which gene is mutated and what kind of mutation it is. And why it is important? Because once you know what is the risk of cancer, lifetime risk of cancer, age-specific cancer risk with different mutation and different gene, which will depend upon your gender and age, you can have early detection through screening, or prevention. Sometimes in some cancers using chemo prevention, but in most cancers chemo prevention doesn't help. So if the risk of lethal cancer is high, then you do prophylactic or preventive surgery. So now I am showing this. I don't want to scare you, but as I said in the beginning, that we have to. So we have to. We have to interpret the reports, and why it is important. In this slide, you show this is a study recent in 2000, in 202 genomic malpractices cases. Payout to plaintiffs in 60% genomics cases compared with 22% plaintiffs in other medical malpractice cases. So doctors are facing lawsuits, and I will show you some examples what is happening with them. And because sometimes they even hold them liable for how they offer, interpret, and counsel patients about genetic tests. It is very important, and this is coming, and it is only a matter of time you will know personally someone who has been sort of taken to the court. And if you don't believe me, I'll show you some cases. So when do you have a serious disease, who has a right to know? So like I'm just showing, this is the UK case just 2 years ago. In the UK, one family dark saga of violence, ill health, and medical confidentiality set the stage for a lawsuit that could change the scope of the duty of care doctors owe to the public. You know, whether you have to tell the family also if something is there. So there are many aspects. So it's not just to the patient but the family. So this lady, she's told a story. Damage for the rest of my life, woman says. Surgeons mistakenly removed her breast and uterus. Can you do that? So what happened? The nurse misread her BRCA genetic test result as positive. That means, positive means not always, positive. BRCA positive means mutation is there. Like, we say COVID positive, it is, one should be negative, not positive. And the gynae and the breast surgeon thought, okay, she has a mutation in BRCA1. Actually, she was BRCA1 negative, and they performed prophylactic surgery. And later on, this lady, when her family was tested and someone was not found, and they looked at her report, they said, well, your report is negative. You don't have the mutation. Why on earth your surgeon removed your breasts and uterus? And then it was discovered. So, and the gynec and the breast surgeon can't say, well, the nurse misread. I mean, you are the doctor, you are doing the surgery, or you are giving a treatment, tamoxifen to someone. You have to know. You can't sort of say someone else. Unless until a genetics team had said that and done that, then you can put the blame on that. Well, the genetics expert has said that, what can I do? Maybe there were some nuances. But if it was anything short of a genetics expert has misread the report or misinterpreted and, mentioned to you, then you have to do that. Like radiologist, their favorite last line is, please correlate clinically. When you see the genetic test results, many times they leave, the ball is in your court. Other test reports, the patient gets the report, but for genetic tests, many times the companies will send the report to you, not to the family, and you have to then disclose the reports. So now I am showing a case. So this is Donna Safer was in 1956, was 2 years old when her father was operated by Dr. George Pack for a colon cancer with
Sir,
extensive. Yes.
sir, the slides are not moving.
What?
The slide is not moving.
Slide is not moving. Is it? Are you?
We're
It's okay.
seeing
The
the
slides are
black
moving,
box.
sir. Moving. It's moving.
Hmm.
Yeah, it's moving.
So it is maybe on your screen. It is not moving. Okay. So, he was a very famous surgeon, Dr. George Pack. He was the cancer surgeon of that era, 1950s and 60s. He had an estate, he used to do many things. So, in 1956, this lady who later sued his estate, not him because he was dead, she was too when her father was operated by Dr. George Pack for a colon cancer with extensive polyposis. Donna's father and Dr. Pack both died in 1960s. Donna was diagnosed with colon cancer and FAP soon after her marriage. She won the case against the estate of late Dr. Pack that her parents were not advised by Dr. Pack about hereditary cancer risk in their children. So just to cut a long story short, the name itself is familial adenomatous polyposis. Of course, in 1956, APC gene testing was not done, but I mean, the name of the syndrome itself is familial. When it is familial, the family is at risk. So, as a doctor's duty, you have to tell. So, maybe he would have told verbally, but there was nothing on record. And because the gentleman who had FAP, he had died, his wife denied that he was ever told. Maybe at that point of time, I mean, any Indian doctors have this attitude. We want to protect the family. We don't want to give them a bad news like, hey, your husband, the wife. We can't tell the wife that your husband is dying, and now your daughter or your son also may develop it later and may have the same fate. So, although on one hand we are trying to protect, but actually we are harming them. Okay, it's like so this is happened. So it is your duty as a doctor, if there is an unambiguous familial cancer risk, to inform the patient. At least if you have informed the patient or someone who is accompanying the patient with the patient permission that this could be hereditary, other members in the family could be there, and you record it, you have done the job. But if you have not, and many times these days in EMR all the records will be there. Your resident may have made a family history that patients, 3 family members have had this cancer, or multiple polyps for that. And if during consultation and follow up that point is never raised only, then it is, and someone takes it up. And mind you, many of them, when I deal, many of the patient has died, but their family members come and then they are very sort of upset that no one ever brought this up. We were not told, otherwise this would not have happened. So when do you suspect? I want to can you see the full slide, Ilima?
Yeah, sir, it's visible.
Okay. So I think all the residents, all the trainees, irrespective which branch of oncology, whether you're radiation oncology, medical oncology, surgical oncology, all of them, I don't know about surgical oncology, but they would have read about Knutson's two-hit hypothesis. And I think if you know Knutson's two-hit hypothesis, you know it explains so many things. Knutson's two-hit hypothesis, and it explains early age of hereditary cancers. It explains multiple or bilateral tumors in hereditary cancers, and it explains family history in hereditary cancers. Whenever there is a germline mutation in a tumor suppressor gene, all these things happen. And of all the genes which can cause hereditary cancer, except RET proto-oncogene, all others are tumor suppressor genes. So, and how he deduced, Dr. Alfred Knutson, he wanted to be a mathematician, but somehow he went into medicine. He then became a pediatrician, then went into a public health. And in his hospital, he was just, we doctors always knew, see, hereditary cancer has been known for hundreds of years. People knew that breast, ovarian, and some other cancers could run in families, even colorectal cancer. So he knew that some retinoblastomas are hereditary because they have a strong family history, and other retinoblastomas have no family history, nothing. And other feature was already known that the hereditary retinoblastomas are more often bilateral, because which are very common sort of which used to go hand in hand. So kids who would have bilateral retinoblastoma, you would more often find that they have a family history. And so then he just plotted the age at which they developed retinoblastoma in the first eye, and whether it was sporadic or this thing. So he took all the bilateral ones as familial and the unilateral ones as sporadic. And then he got, if you those who have studied radiobiology, the linear quadratic. So it was a two-hit hypothesis, like you have the cell kill two-hit hypothesis. So he deduced that in some patient, like those who have a hereditary type, they require just one hit, whereas those who have sporadic type, they require two hits. And because ultimately the disease was same, retinoblastoma, from that he deduced that the hereditary ones are born with a germline mutation. First hit they already have, whereas in the sporadic one, the first hit happens after they are born, which is a somatic mutation, and the second hit, so the hereditary ones require just one more hit, one additional hit, because as I said, all the genes have a pair copy, one from your mother, one from the father, two alleles we have. So in the hereditary one, the second hit happens after they are born, so they only have to have one hit, whereas in the sporadic one, the same cell has to undergo two hits. The same gene has to be knocked out, both the copies of gene has to be knocked out, so that that gene doesn't function. So as a result, what will happen? Because you require only one event, here you require two events, it will happen earlier, that means younger age. And because this first hit is present in every cell of the body, more likely it will be bilateral, more likely it will be multicentric, and or multifocal. And sometimes because of the germline mutation present in every cell of the body, they may have other phenotypic features of that syndrome. And obviously because it's the germline, if you have got it from your mother or your father, there's a high chance that your sibling also may have inherited this. So if you remember the Knutson's two-hit hypothesis, it explains everything about when to suspect hereditary cancers, a cancer which is occurring earlier than the expected age, a cancer which is bilateral or multifocal or multiple primary cancers, and when there's a family history. So, like in everything else in medicine, we make syndromic diagnosis and differential diagnosis. So, differential diagnosis is very important in a genetics clinic. If you don't make differential diagnosis, you will be lost. You never get a classical hereditary, cancer. Sometimes you get a classical, but most of the time you make a differential diagnosis, and that is based on the type or the histology of the cancer, age at cancer diagnosis, whether it was unilateral, bilateral, or multiple primary cancer, histological or IHC features, who all are affected in the family, does it follow a Mendelian inheritance pattern? Many times, one patient said, I have a family, say, who said my mother-in-law had cancer. So mother-in-law, of course, she's your family, she's like your mother sometimes, but she is not your blood relative. So in the blood relatives, how it is being inherited. And one thing I don't get surprised now, even doctors, that's how we take history, and then we even explain. They say that, but BRCA1, why do you have to test in men? Can it come from the father side? So I tell them it is not a sex linked, this thing. So that's why I say that, although it is, one shouldn't be telling this such a basic thing, but so many people are confused about it. BRCA1, the penetrance is more in women. That means if there are 100 carriers female carriers of BRCA1 mutation, 70, 80 of them will develop breast, ovarian, or other cancers. But if there are 100 male carriers, only about 20 of them will develop prostate cancer or male breast cancer. But it is on chromosome 17, which both men and women have. So it is, you have to follow. So like typically in hereditary breast ovarian cancer, if a lady has breast cancer, on a mother's side, there will be no family history. Her father may be unaffected, but her father's sister may have cancer. Or her father's brother's daughter may have had cancer. So you would say there's no first degree, but it is because of the differential penetrance. Then the other phenotypic features are very important, as I mentioned, that doctors are phenotypers, polyps. Skin is the largest organ, which is you can actually see, and so many syndromes would have cutaneous manifestation. Okay. Even in our dark Indian skin, we can see cutaneous manifestation very clearly, provided you look for it. Eye is the only internal organ which you actually see with a fundoscope. Unfortunately, these days, most outpatients don't have a fundoscope. I hope the neuro OPDs have. Even the wards don't have a fundoscope. Even if they have it, there's no cell, you can't operate it. Cysts on imaging, if you find in kidney, in liver, or in pancreas, or something, they may be a telltale sign of certain syndromes. And in the brain, there are so many features which are very characteristic of a particular syndrome, which I will not get into details of it. So, I'll just talk about, DNA, genes. When genes are changed, then amino acid and in genes, the amino acid in a sequence make proteins. Proteins have biological function, which is normal growth and physiology. So, any spontaneously or due to carcinogens, the DNA sequence is altered. Because of any reason it is altered DNA sequence, it is a mutation, technically speaking. So genes with genetic alteration or mutation, only some gene mutations alter the structure or shape of their coded protein. So many gene mutations, what we call synonymous or silent mutations, which are very common polymorphism, they don't change it. Sometimes even if the amino acid has changed, but the function or the structure of the protein hasn't changed, and ultimately proteins are the effector molecule. So nothing may happen. And even some protein alterations change their biological function. That means that some alterations in protein functions are also tolerated, or they may be redundant mechanisms. So every genetic, and ultimately what you are doing in genetic testing, you are just seeing whether the DNA sequence is altered or not, and rest of it is all interpretation. So even some changes in biological function, only some changes in biological function result in a disease, congenital anomaly, or evolution. That means many biological function which are changed, which are altered, but there's a redundant pathway. There's something else. In the metabolism, we know there are so many, phase 1 enzymes, phase 2 enzymes. If this GSTs are not working, something else is working. If SALT is not working, something else is working. So there's a lot of, scope or reserve in the human physiology, or the, not just human, all physiology. So every genetic alteration, every mutation is not a cause of disease. And I think that's where the whole trap is, because finally you have to interpret the genetic testing report in terms of whether it will cause the disease or not, or whether it is the cause of the disease this person has, and whether you should act upon it. So this is what we call variants of uncertain significance, VUS. So when you do genetic testing, you will find very many VUS. I will talk about it later. India is a subcontinent. This is a geological entity which has been created over God knows how many millions of years ago. And it's also a socio-political entity. But people have sort of moved here and there. So essentially, it is a one large gene pool with several thousand smaller gene pools within this, of few 100,000 to few million people. We have 1.5 million new cancer cases, of which, even if you take only just 3% being hereditary, we have about 50,000 hereditary cancers. Unfortunately, there's not a single comprehensive cancer genetics program in South Asia. Ours is also not comprehensive. I mean, it is near, closest to being that. I know what are the deficiencies in our program. Now we see in Tata Hospital about 5 new families every day, hereditary cancers, and 10 follow-ups. Now we have 9,000. So this is the growth, how it has happened over the years. Once upon a time, it was my lunchtime clinic. Now our genetic counselors don't have time for lunch. That is the scope. So this is just a snapshot of the mutation that we have found in our lab. So we have now more than now it is close to 2,000 individuals and about 1,200 families in whom we have identified a germline pathogenic mutation in one of the 18 genes that we test in our lab for hereditary cancers. See, the real beneficiaries, so the red ones are the ones who have like this BRCA1 mutations. These are the relatives, 290 of the relatives in whom we found a mutation. They were as yet unaffected. So they could be advised about prevention. The real beneficiaries are the ones mutation negative relatives. So they were living in the fear that, like my mother, like my sister, I will also develop cancer. But on testing, we found that they were lucky, the 50-50 chance, flip of a coin, they did not inherit the mutation. And there are many other genes. So I won't go into details, but because Indians used to marry within their region and sub-community and sub-community. Religion is a very broad thing. Within that also there are so many things that if a mutation arose just by chance as a probabilistic event, germline mutation, say 1500 years ago, it is highly likely that today there will be many people of the same community, the person in whom the mutation arose, they will have that mutation, what we call founder mutation. So we have many founder mutation They're in different parts of the country and in different communities, and in BRCA1, BRCA2, MLH1, MSH2, and some other genes. So how do we get about it in real life? So who should be counseled and tested? So when someone has a likelihood of inherited predisposition to cancer, uh, traditional cutoff people used to take 10% probability, but there's no consensus on that, especially when you're paying for yourself. NHS may use a cutoff based on what NHS can manage to do, but here everyone is paying for themselves or through research. Therapy, when there's a personal perception about inherited cancer risk. So again, one thing is important. As someone, you may as a doctor may think that this patient does not require genetic testing. Cancer doesn't seems to be of the genetic type or the hereditary type. But if the person has serious concerns about it, he or she may still require genetic counseling. So genetic counseling is distinct from genetic testing. After genetic counseling, someone may need genetic testing or may be told, well, you have an option, or someone may be told, in your case, unlikely genetic testing is going to help in any way. So that is the one thing. Then what are the options available if a mutation is found? So really speaking, about 10% of all cancer patients would be eligible for counseling and then testing. But in real life, it doesn't happen. So what we have done many things to reduce time, effort, and cost of testing. But all the testing we did, I showed most of the data except for some who got testing done in private labs. We relied heavily on phenotyping, pedigree analysis, and syndromic differential diagnosis. So by doing that, we could zoom in and reduce the number of genes and the hotspot that we could test, and we had identified many founder mutations. There are a lot of ethical, legal, and social implications. So what we call LC, it's a minefield. It's a double-edged sword. Genetic testing would actually harm, and I've seen patients afterwards, they have come to us because they had not been counseled about it. The patient did not know that they have undergone genetic testing. When the report came, it was hidden from them, and so many things. And unfortunately, young girls, the parents, sometimes there must be many young women in the group. I should say that every young woman is not considered as an adult by their parents in this day and age, especially if they haven't got married. Women will trust their parents. Parents also love their children for them, but they have got genetic testing done and they did not know, and their blood sample was taken. Okay, let us do something else for maybe this thing, and that was sent to genetic testing in lab. And when the mutation came, then now what do we do? Because when mutation is found, you have to counsel them, you have to explain to them something. So this happened. So the myth is there's one large family where everything is shared. It is a myth. Each family and each person is different. There are many common and some unique issues. And as I've told you, what are the issues? So phenotype, I've already mentioned about phenotype and genotype again, and why I want to show this, that you don't look at the low risk SNPs. Some genetic test reports are so misleading. They are reporting SNPs as the, as a mutation, and some doctors are acting upon it. Someone is giving PARP inhibitor, someone is referring for prophylactic surgery for SNPs. Just ignore them. Somatic mutations can tell you about something else, but they are not actionable for the family, and there are many other things. So now coming to the colorectal cancer, I've explained most of the basic things because unless until you know them, just talking about colorectal cancer genetics makes little sense. So we know the etiology, factors, and pathogenic is a complex. Medical history is very important. Family history, dietary factors, we know lifestyle, and for the hereditary type, it is the family history and the pathology features and the endoscopic findings which are the main thing. But patients ask about the diet, so you should know about it. So about 5 to 8% of all colorectal cancers have a family history and clinical features suggestive of highly penetrant Mendelian CRC. So this is like a hereditary colorectal syndrome. Another 10% are familial. They have family history, but not like that of a Mendelian disease. And the remaining are sporadic, that means no family history, but even sporadic ones could be early onset or they may have features suggestive of this thing. So how do we go about it? First and foremost, detailed evaluation to distinguish between sporadic, familial, and hereditary cancers. And for suspected inherited predisposition, then you have to do a very detailed evaluation of colorectal and extracolonic manifestation with clinical, endoscopic, imaging, and histological examination, and then based on that you say whether it is likely to be hereditary or not. If it is hereditary, whether it is a polyposis type or non-polyposis. And once you suspect it is polyposis or not, then the histology is very important. So we all say polyps, but polyps could be adenomatous polyps, they could be hematomatous polyps. Of course, there could be some other types of polyps. I won't go into detail, serrated and all that, but mainly it is adenomatous polyp or hematomatous polyp, and the number of polyps. So if it is just 3 polyps, 5 polyps, 40 polyps, or you can't even count them, carpet of polyps. So even in when we say the name syndrome, I've written it here, Lynch syndrome or hereditary non-polyposis colon cancer. This is an old name because this is a misnomer. First of all, many patients with HNPCC actually may have polyps, so it is not non-polyposis, but their polyps may be 3, 5, 7, 10 over a period of decades. Whereas in FAP, familial adenomatous polyps, there will be a carpet of polyps. Then, so based on the polyposis, what kind of polyps, number of polyps, adenomatous polyps or hematomatous polyps. Adenomatous, if it is autosomal dominant, that means you have a family history, father also had and no one else, and you have, then it could be FAP. But if suppose there is no family history, but 2 siblings have, suppose colorectal cancer and multiple polyps, but their parents give a history of consanguinity, or they are from the same small community, then it could be autosomal recessive, which is MYH associated polyposis. In hematomatous polyposis, it could be Pewds Jagger syndrome, it could be Cowden syndrome, it could be juvenile polyposis syndrome. In non-polyposis, the most common is the autosomal dominant Lynch syndrome and the autosomal recessive CMRD. Again, if autosomal recessive, you may not get a clear-cut family history, but I will talk about it very briefly later. So this is a spot diagnosis, those who can see it very well on your screen. So these are the 2 cancers. You can see again, Knutson's hypothesis, 2 cancers, 2 multiple primaries. You can see ulceroproliferative necrotic growth here. And you can see these are polyps. Okay, polyps are not always hanging down on a stalk as you would read, like this thing. Sometimes this could be polyps. So this patient, this is just 20 centimeter of resected colon, has almost 100 polyps and 2 cancers. So it's a spot diagnosis is, I don't have to say that, FAP. So this is a diagnosis which I'm sure Dr. George T. Pack made, but he did not take effort to tell the family that it is a familial condition. He just treated the patient. He did not advise between treatment and advice. These are 2, it is overall management includes advice to the patient and the family, which he did not do, and his estate had to suffer. I think they had out of court settlement afterward. That's what I remember. So FAP, it's a rare autosomal dominant, accounts for less than 1%. Penetrance is nearly 100%. And there are 2 variants. One is attenuated FAP, depending on where in the APC gene the mutation happens. They have a fewer adenomas a little later. So they may have 50 or 60 or 70 polyps, not few hundred. And the other one, as I said, MYH associated polyps. Now, FAP has a lot of extracolonic manifestation, and sometimes it may happen that the patient comes to you with that particular extracolonic. So if thyroid cancer, they may have papillary thyroid cancer. Brain tumor, generally it will be a medulloblastoma, hepatoblastoma, jaw osteomas, desmoid tumor, congenital hypertrophic retinal pigment epithelium. So patient may present with any of these features. So APC gene, it's a long gene, spans 15 exons, and it's a tumor suppressor. And MYH is another gene which is on a different chromosome 1p. Again, it's a tumor suppressor gene. And this is APC is involved in the Wnt pathway, whereas this is involved in the base excision repair pathway. Okay. So I won't go into details of it. In the APC gene, depending which region of which exon of the APC gene has the mutation, the phenotype may be different. Surgery, again, I don't think there are too many GI surgeons, but again, all I would say, this surgery should be planned and done by someone who has a lot of experience of dealing with such patient. Of course, not of experience doesn't mean that person would have dealt with few dozens, but at least would have read and dealt with 1 or 2 or in the unit or consult, because Other thing I did not mention, they are prone to develop fibromatosis, and sometimes they die because of extensive fibromatosis, and that sort of traps the ureter, that traps, really messes them up. So they have tendency for fibromatosis. So that is one thing. And main thing is, the issue is how you can avoid a permanent colostomy in them. So depending upon the lowest extent of the polyps, and if suppose you have saved, so you have done ileorectal anastomosis with J pouch, keeping a very close watch on the remaining part of the. So we have published our data. I have 2 PhD students, so this is only Indian data which has been published. That time there were some 56 families with FAP. Now we have close to 70 families with APC. And as I said, it doesn't stop. If we don't find APC, then we found MYH, then other genes are there which could be screened. And majority of the mutations are nonsense. That means the protein doesn't function. There could be a mutation known as large genomic rear engine, which will be missed on your routine genetic testing. So what is a technique known as multiplex ligation probe amplification, MLPA, it's done a different way. So if you have a clear phenotype of FAP, if you have done for NGS and the report is negative, don't say nothing, it's not. You may order a MLPA test, because in FAP, the clinical phenotype is unequivocal. Nothing else can produce that phenotype. MYH I have mentioned, I will not go into the details. So this is a pedigree which is had 2 heterozygous germline mutation. These are other genes which we do for research. If you don't find one odd case, so when you have students in the lab who do PhD, they have to do research. In the research, the focus is, if someone has a phenotype like FAP, and if you don't find mutation in APC or MYH, could there be something else? So then we did a germline whole exome for them. We didn't find anything. So, but in India, many patients will not be able to afford. So this is my student, we published this few years ago. So even just with simple, by amplifying and doing sequencing of 5, exons, you can identify majority of the mutations in the APC gene. In Lynch syndrome, which is HNPCC, the mutations spectrum is different. Very few or no polyps, early but variable age, and most tumors are right-sided. As I've shown here, okay, in this, if you see, normally most of the colon cancers or GI cancers are mostly on the left-sided, but here they could be more of right-sided cancer there. And they are mucinous signet ring cell morphology, and they could be synchronous or metachronous cancer. And this again, autosomal dominant, high penetrance, and there could be many mismatch repair genes. And I am not explaining the mismatch repair pathway, which could be MLH1, mostly these two, MLH1, MSH2, or else MSH2, MSH6, and sometimes PMS2. These are the main genes that will be there. And extracolonic cancers, they don't have so many as in the FAP, but very important, endometrial cancer is a very common. Half the women with mutation in this gene will develop endometrial cancer. And generally, they will be high-grade endometrial cancers. Stomach cancer could happen. Brain tumors in Lynch syndrome are not medulloblastomas, but they are glioblastomas. Hepatobiliary cancers, pelviureteric junction cancers. They are not so rare, about 5-10% patients. Ovarian cancer, small bowel, and sebaceous gland. So especially the Muir-Torre syndrome, when you have sebaceous tumors. So normally we ignore sebaceous, or if you have any skin this thing, but if someone has a family history or a personal history of colorectal cancer, endometrial cancer, if a skin lesion is growing, they need to be biopsied, because it could be. And the Turcot syndrome is basically brain tumor glioblastoma with CRC. So the clinical, I'll come to the main point. So if after family history, there are many Amsterdam 1, 2, then revised Bethesda criteria in Amsterdam. So most people use these criteria, but in revised Bethesda criteria, every second patient with colorectal cancer would sort of fit into it. Because if you say anyone who's diagnosed with CRC is under 50, half of our patients are under 50. So what I'll tell you, what we do in our clinic, we rely heavily on IHC and MSI analysis. Of course, family history and phenotyping we do. So microsatellite instability is It's a hallmark of Lynch syndrome tumors. Regions of DNA which are composed of short tandem repeats. So like A, so 80, 5 times, or something like that, 20 times. So, and they are stably inherited, repeat length same in every tissue. And in microsatellite instability, small deletion or expansion within the short tandem repeats in the tumor DNA as compared to the matching normal DNA. Because tumor cells are multiplying, so it is like when you try to say seashells on the seashore. When you have to repeat many times fast, you are likely to make a mistake. So same way when the tumor has to multiply many times, so parts of the DNA which have a repetitive element like 80 or ATC, they are likely to, one ATC may be deleted or added. So if so these are the Bethesda NCI panel, 1, 2, 3, 4, 5 markers. MSI high, we say if 2 or more markers are unstable, that means they are changed from the original. MSI low, if it is instability is only at 1 marker, but it is not there, and stable if it is all the markers are stable. So this I am showing, so this is the DNA from the lymphocyte, that means the germline. Lymphocytes multiply normally, they are never under stress to multiply very fast. And this is the DNA from the tumor we have extracted. You see, this is what we call fragment length analysis. So like that particular BAT 25, this marker, suppose it was 80 repeated 18 times, okay, 80, 15 times. So you see only one, this thing, it is a homozygous. In the tumor, you can see that there are two species, that means it has shifted, that means it has become unstable, it has changed. So here also it has changed. So this is on fragment length analysis. You're looking at the repeat lengths of the two alleles have changed or not between the tumor and the normal cell. This is on IHC, and IHC what we see, the loss of expression, like MSH2, MSH6 go together because that is how they are, this, and MLH1 and PMS2 go together. So using internal control, so the expression, these proteins are expressed on all cells. So lymphocytes, they are expressed quite well. Normal glandular epithelium also, they are expressed, these proteins. So we use them as internal positive control. So if they are expressed MLH1 on the lymphocytes or on the glandular epithelium, but it is not expressed on the colon cancer cell, then you say it is loss of MLH1 expression. Okay. So here MLH1 expression is lost, and PMS2 also goes along with this. So in this patient, we would suspect this patient has a loss of MLH1 expression in the tumor cells. So maybe due to mutation in the MLH1, and so this patient will require genetic testing. So we have very little data. I'll just show you the okay, I'll just show you the details. So we had 159 suspected. Yep. Someone is saying something? Okay, so we have 159 suspected Lynch syndrome cases. If you see IHC status, we knew in about 117. And mind you, this IHC has to be done in a proper, so our is a CAP accredited lab TMS pathologist. So they are expert GI pathologists to do that. And which antibodies you use, are you using a diagnostic antibody or something just for research, and you're using internal controls? And how good are you in interpreting that? Sometimes we say the result is uninterpretable. You have to accept it. Instead of just giving some result, if you cannot interpret that result with this thing with confidence, you should say it is not interpretable because internal controls did not work. So in 72 patients who had a loss of MLH1, along with that PMS2 also goes, we found a germline mutation in 67 patients. Similarly, in patients, 43 patients who had a loss of MSH2 and MS6, like I showed the case before, we found a mutation in 43 patients. So, and when you don't have a IHC status, was based on family history also, because family history, of course, is the most important thing. Before IHC, we were relying only on family history. You will still find mutation, but of course not in all. And the mutation spectrum that we have got in our families, which is quite reasonably well similar to the Western population, Caucasian. Majority of them have mutation in MLH1 gene, then MSH2, then MSH6, and then really PMS2. And then these are many founder mutations. I'm not getting into details of the founder mutation of one. Now we are just sort of, time is over now. So I will not get into the founder mutation. This haplotyping I will not to get into that. I'm just showing you a few classical HNPCC or Lynch syndrome families. I used to myself write it HNPCC, but I would. So you see early onset colon cancer, multiple siblings affected. So this lady is unaffected, but her son has colon cancer, so incomplete penetrance. So she may develop the colon cancer or endometrial cancer later, and a father had, and these are the kind of histories that you will get. Again, this is a classical HNPCC family. So like here, we have found that unaffected people, these are the ones we found the mutation, they're as yet unaffected. These are ones whom we will actually help them by doing on lifelong screening and early detection. And for Lynch syndrome, you don't do prophylactic colectomy. So you just keep them on lifelong screening. So this is the penetrance. Oh, it's very important. A lot of people used to ask, okay, well, maybe Indians, our diet, we have more of curries and curcumin, so we won't get colon cancer because vegetarian, even if we have mutation in MLH1. But unfortunately, when the mutation is in a gene like mismatch repair gene in MLH1 and MSH2, lifetime risk of cancer is quite high, close to sort of 80% in our cohort. And for colorectal, almost same. And for MLH1 and MSH2, more or less similar. And this is for males and females I'm showing later, because important to see in endometrial cancer risk, in MSH2 carriers, this is close to 60%. MLH1 carriers, it is close to 40%. MSH2 gene, one thing I would like to say that this has more of extracolonic cancers. So, when you have, so whether it is a skin cancer, whether it is ovary cancer, endometrial cancer, prostate cancer, they are more in MSH2. Of course, in MLH1 also they can happen, but they are more common in MSH2 gene mutation. Endometrial cancer is already. Even breast cancer now we know is risk is increased in patients who have germline mutation in the MMR gene. And for MSH2 especially, the lifetime risk could be as high as 20%. So now Pewds Jagger syndrome, I just want to show that this again we have published last year, another PhD student of mine, that STK11 gene, and the phenotype of that was not very well. Of course, from India there was not a single case. So classical PJS will have cutaneous pigmentation and on the lips, inner side of the lips, and hematomatous polyps with or without family history may not always be there, and that hematomatous polyps may cause intersusception. So the family history may be of childhood acute abdomen and intersusception, and we found that mutation in many cases. And in breast cancer, what we described for the first time, they have a typical phenotype. They are ER/PR positive and HER2 negative. CMMRD, which is constitutive mismatch repair deficiency syndrome. It's a rare syndrome. Actually, it may not be that rare, but it is often missed in the clinic because this syndrome was never described earlier. So there's a CMRD consortium world over. There are only about 150 families described. So basically, you may have a Lynch syndrome spectrum tumor at younger age, multiple polyps in absence of APC mutation, and high-grade glioma, which is very common, lymphoma, leukemia, typical, leaf from any kind of cancers, and they may have a consanguinity. So based on that, what we found, we have found mutation in 4 families or 5 families, and that sort of confirms the. So PMS2, the mutations in CMRD, the most mutations are in PMS2 gene, not the MLH1 or MSH2. The testing for PMS2 is a little tricky. It may be, you may miss the mutation because it has a pseudo gene which looks like PMS2 sequences, similar sequence homology, but it is a pseudo gene. So in our lab, we developed a technique which is not, we standardized a technique known as long range PCR, nested PCR, and we found mutation in many sort of families. And this is a pedigree. You see consanguinity is common in this CMRD family. This is a mutation. So from our study also, we find the 3-point scoring system is validated. So this paper we are in the process of sort of communicating. And I just want to warn that NGS, because of the pseudogene, many times it will give you a false positive. Mostly it is false negative, but for PMS2, if you get someone is shown has a PMS2 gene mutation, actually it could be a false positive. So don't act upon it in a serious way. We'll be very happy to receive your sample and confirm. Now we are more and more people are using NGS, makes life easy, many other things. But one thing it is happening, I'll just show one last data slide, that in many more and more genes you test in a population which has not been tested earlier, you find in about 1 in 4 patients. It was data about 2 years ago. Now we find 1 in 5 or 1 in 6 patients, we find a variant of uncertain significance. So when you find a variant of uncertain significance, in many patients, you just tell them, don't worry, they will stop worrying. They will just listen to you, that no actionable mutation. But in some of them, because there is something written and it's a variant of uncertain significance, and when doctor explains to them, well, I don't know what it is, but they stop worrying. So the counseling of VUS is quite difficult. But over a period of time, I think our VUS rates will come down. So this I'm showing, this is a VHL family. So writing is on the wall, now patient knows it all. This is the patient, this patient's brother-in-law. After everything else, he said, oh, sir, we know that. In VHL, we have to look at this. I said, how do you know? He said, sir, we have our group. He flashed his phone. Then with his permission, I took his photo. So he said, when we come to Tata Hospital, doctors say we get tense, we put in our group, it is all clear. We are happy. Okay. So now patient is knowing it all, and how they know it, because every doctor tells them something different and they figure it out. It's not matching. So it's the snake and ladder of genetics. It was like a poison, snake's poison, which was running down generation. But with genetic counseling, genetic testing, you can heal it. You can heal it. People can go through the journey of life. And if you have any patient, if you can't deal with that or whatever constraints are there, we are happy to provide telephonic counseling or genetic testing in Homi Baba block, 3rd floor we are, and you have my number. And I would like to thank ICMR and DBT, my lab, Dr. Pradnya, Ravi, genetic counselor, and I had 2 PhD students who have finished and written their PhD in few papers, and few more papers are coming on the way. Thank you.